The idea of today is to give a comprehensive recorded tutorial of everything that MSM is about and what you can use it for. And so to kick it off, I thought it would be useful to give a sort of potted history of why MSM is the way it is. Like what was it originally designed to do? And that goes a long way to explain why it's designed the way it is. And then I'll go into like high level detail about how it's optimized, just to give a bit of context to what all of the configuration parameters mean. So MSM was originally developed when I was working for the Human Connecting project, and the goal that I was given was to develop an image registration algorithm which would improve overlap of cortical functional activations across all participants in the HCP. Where, as we all know now, the long-term view is to develop the human connecting multimodal cortical aerial parcellation. But the short-term goal was to improve overlap of functional activation so that we could do more sensitive task-based analysis with fMRI. And so at the bottom here, can you see my, yeah, at the bottom you can see this is an HCP participant. This is a movie of their resting state image, and hopefully you can see that even though this is, you know, or at the time of state of the art in terms of acquisition image processing, this data is still quite noisy. So this was the space that we were working in. And so jumping ahead of myself a little, which I'll do a lot in this presentation, the overall goal here basically was to improve overlap of these functional activations that you see popping up in here, so that when you did some task-based analysis and then you ran a general linear model on the mean activations across the population, so you're looking for areas of the brain that commonly activate across all participants in a task, you pick up more detail basically. So the better overlapping the the activations are across the participants, then the more likely they're going to survive statistical significance thresholding. And so you're going to see more clusters, and you're going to see you know, greater overlap of clusters across participants. So this is the starting point. This is where we started with FreeSurfer, and this is where we ended up with MSMall. And you can see that in terms of the cluster mass, which is a measure of the extent and the intensity of these clusters, you get an enormous boost when you run image registration with resting state networks. And, you know, just to point out throughout that this task-based analysis, at no point is task ever used to drive registration. It was resting state fMRI. So this is an independent evaluation that the cortical areas are better overlapping after MSM. Okay, so this is where we started out. This is where we ended up after the first paper. And I'm going to talk a little bit about why getting to this point was actually really hard. So to understand that, we have to talk a little bit about how people classically do neuroimaging registration for brain scans. So historically, what people have always done is they've performed their image registration on structural MRI volumes. So the objective was to just smoothly to form some input volume until the structure in this image, so the cortical folds, the ventricles, the subcortical structures, overlap better with that of some target or reference volume. And on the face of it, that sounds straightforward. You know, we're nicely, smoothly deforming this. It looks reasonably good by the time you get to the end of it. But actually, in reality, when you really look at the detail, and it's a lot easier to do this on the surface, so these are in flirty surface maps, there's a huge degree of cortical folding variability across participants in, you know, in the normal healthy population, and Yourong will talk a lot more about this. So you often see that you'll have two folds in one subject where you have one in another. You'll have different orientations of folds. You have different branching and splitting of folds, and there's simply not enough information in the single grayscale image to tell you how you're supposed to split two, you know, one fold into two. It's just literally it's just guesswork. because it's based on just this grayscale information. And so that means that this registration problem is ill-posed. And so in order to learn something that is reasonable, most often you impose a number of constraints. So firstly, most image registrations attempt to learn what are known as diffeomorphisms. So that means that your image registration needs to be smooth and invertible and continuous. So neighboring points are smoothly moving in the direction of their neighbors. There's no swapping over, there's no discontinuities. And that makes sense because that means that you can't just take any point in the moving surface and move it to any location in the target surface. And this may seem obvious and not what it would do for structural, probably not, but actually this is exactly what happens if you drive functional registration in a completely unconstrained way. So this is bad. So imposing diffeomorphisms makes sense, so you don't learn something completely insane. And the other thing you'll do is you'll impose some regularization on how smooth that diffeomorphism is. So that will place some first or second order constraints on how neighboring points or groups of points move together. So we don't want any area to stretch or compress too much. So this is how image registration classically works. And this is which is still most popularly done often, is that we drive registration from structural volumes. Okay, so the problem is that most of the functional, if we're interested in high order cognition, then a lot of the functional activations, in general, a lot of our functional activity is occurring in the cortex because it's occurring in the grey matter tissues, which are within the cortex, this thin layer of tissue at the outer surface of the brain, and within the subcortical grey matter structures. And the cortex is a highly convoluted surface. So it doesn't make sense to represent it as a volume. And in the context of image registration, this is particularly pertinent because what happens is that you could imagine that you have this pink gyrus and another subject has this green gyrus. And this green gyrus is slightly shifted with respect to the pink one. And if we were doing image registration in volumetric space, this small shift, this small error, this small deviation in where it's supposed to be, is not going to have a strong penalty. However, if we were to do it in if we were instead to consider our distances along the cortical surface, then the true distances between these points here are really long. And if we were trying to register the right hand bank of the sulcus to the left hand bank of the pink sulcus, then what we're going to do is compress all of this information into nothing, basically, and that's not going to be allowed. So in the context of image registration, representing the cortex as a surface, which it is, and penalizing or regularizing deformations based on geodesics along that surface is going to much less likely lead to anatomically non-plausible deformations. And so for these reasons, FreeSurfer in the late 1990s developed an image processing pipeline that would get you these surfaces out from a structural volume. So you're fitting an inner and outer cortical surface to the structural MRI volume. You then inflate them because this gives you better visualization of your data, so you can see inside the insula. And then from the inflated surface, you project to the sphere, and then you learn mappings on this simplified 2D spherical surface. So this makes sense because we're no longer trying to learn complex sort of displacements between complex shapes in 3D. We're learning how to move points along this smooth 2D spherical surface in order to align these functional maps. So these represent spherical sulcal depth or core scale convexity. So in the blue here, you've got the targets of your image registration. So this is their sulcus. And in the green and red, you've got the moving mesh. And all you're doing basically is moving along the surface, pulling these green bits out so that they fill in between these blue lines. So that's all you have to do. And the other advantage to spherical alignment, which I suspect I won't remember to mention later, so I'm going to mention it now, for functional alignment, is that by completely smoothing out all knowledge of the original shape, then we can deconfound this issue of shape when we're trying to register function. So this makes sense on a number of levels. And this was shown with independent value validation that was run on post-mortem data to improve the overlap of cortical areas. So they took a bunch of dead people and extracted their brains and performed histological dissection to get out cytoarchitectonic areas or functional areas. They then ran image registration in the volume and the surface and looked at how well these areas overlapped in volumetric, which is the black bars, and surface-based registration. And you see that there's considerable better overlap of these cortical areas after surface registration in primarily in primary cortical areas for reasons that, again, Yourong will go into in more detail later. Basically, as you get away from primary areas, the degree of cortical folding is then going to make it more difficult. But in the context of just comparing shape-based registration for surface and volume, then surface is better. So fine, why not just use FreeSurfer to align folds? So the problem here is that folding patterns do not align with function across most of the brain. And I did a quick Google to see if I could find any good image examples from like the large body of evidence that existed before the HCP multimodal partellation, but I couldn't find them. So again, being a bit circular, I'm just going to show evidence of what happens when you run registration with MSM. So if you drive registration with function, then you generate average maps of the language. So this is a resting state group, the average of the resting state maps for the language areas, and similarly for the task, the language task, and you've got T1 over T2 weighted Milan maps and another RSN here. And you can see that in the case of all of these aerial features, as we call them, so features that are aligned with the aerial organization of the brain, then image registration generates sharp group average maps with strong boundaries that look very similar to the individual subject maps, admittedly with less noise. But if you look at the folding map, then when you've driven registration with function, you lose all of the information about folding, which is a fairly strong indicator that your folding is not overlapping with your function. So if we drive registration just with FreeSurfer and then we try and do functional analysis, we're not going to get as much overlap of our functional activations as we possibly could. And Matt Glasser and the Human Connecting Project team, when they set up and asked for MSM, they already knew this was going to be the case. But this is the visual evidence of that. Okay, so you could say, all right, well, fine, let's just use FreeSurfer to register function. This also doesn't work. And to understand this, you have to understand something about the FreeSurfer optimization algorithm, or the FreeSurfer cost function. So if we consider like image registration generally, or in fact most image processing, you have some driving term, some data similarity term here, whose objective is to drive registration in the direction that's going to improve overlap of equivalent structures in the source and target meshes. And then, as we've already said, you have some regularization penalty here, which ensures that we don't just move these points around anywhere to optimize similarity. We do it in a way that's biologically plausible. And then most image registration algorithms optimize these using continuous optimizations techniques, where FreeSurfer uses the simplest of these, so gradient descent. So there, what you're doing is you start off with you measuring the overlap of your image at some starting location. And then you take some steps. So you move your mesh, or you move the vertices of the mesh in some direction that's going to improve the overlap of features between the moving and target mesh. And you do that incrementally. So you take little steps until you reach the minima of your cost function, where always these regularization terms are contributing to that cost function. So your overall cost, you're moving in a direction that's going to improve similarity whilst not creating too many distortions, and you're moving step by step in a direction until you get to what you hope would be a global minima. FreeSurfer does this in a relatively simple way. So first of all, when we talk about deforming the mesh, we're always talking about deforming the native mesh, the high resolution meshes. So you're working at very high resolution, which means that your vertex points on your mesh are very, very close together. And that immediately creates problems because there's too many points that have to move together with each other. And the regularization that it's imposing is relatively weak. So the data similarity cost that it's using is a MSC, so the difference between the sulcal depth maps of the input image and some mean across the population. And this is the variance across the population. And it applies some smoothing kernel to deal with all of this intersubject variability. And then it opposes regularization in two ways. They have a metric distortion and an aerial distortion. So your aerial distortion says don't stretch or compress the triangular faces of the mesh too much. And the metric distortion says let's not change the relative distribution of the neighborhoods too much. This is the first order penalty. It's just looking at pairs of neighbors. And this would all be fine if there was a global minimum, but there absolutely isn't. We've already said there's like lots of pockets of good alignment. You could think that if you were going to align two folds to one, then you could just align one fold to one of the two, and then it'll reach a local minimum and it'll go, this is great, I'm going to stay there. What are you going to do with the other one, for example? So you never have a simple, smooth, convex optimization function. You have lots of pockets of little minima, and gradient descent is going to get stuck in those local minima, and it's not going to be able to get out. And it's a particular problem with FreeSurfer because it's working at high resolution, so it's trying to move too many close together meshes and get them to agree, and that's a problem. And the regularization isn't very strong, so it's not doing a good job of getting them to all come with each other. And so what happens with FreeSurfer, when you actually look at the distortions that it's learning, is it's just shifting points close to boundaries. You can see that these are like, these red lines are bordering the motor and sensory gyres, I think. It just moves things a lot. So it's not actually that smooth across the surface. It's just moving things close, very close to the borders, because it's mostly to do with this use of high resolution, estimating a deformation field straight for the high resolution mesh. Yes, I've already said that. Oh, but the main point that I want to make is that this becomes a much bigger problem when you're talking about aligning fMRI, because the fMRI is super noisy. So the pockets of noise mean that there are a whole ton of places that you can get stuck in a local minima. So you could register activations to noise, you could register different points of noise which correspond to completely different things to each other. It gets stuck. So I spent a long time trying to get gradient descent based methods to work on multimodal alignment, and they did not work at all. So this is why it doesn't work. So the first solution, which is common in volumetric registration methods as well, is that you instead frame your registration in a course-defined way. So you learn, you create control point grids. So in the volume, they're low resolution Euclidean grids, and in the sphere, they're these icospheric grids. This improves alignment because it gives you more freedom to move smoothly. You can learn a larger, smoother deformation at a low resolution, and then you can refine the fine detail as you upsample your deformation grid to finer and finer resolutions. Because even if your regularization is relatively weak, this point here can easily move here without switching or swapping over its neighbors. So it's got more space to move, so it can move in a bigger way. But if you're working at this resolution, then you're only going to be aligning coarse features, so you have to refine by going higher to higher and higher resolution. And the other thing you can do is use a better continuous optimization scheme. So this is what Spherical Demons does. Basically, it implements optimization. So it implements optimization in a coarse-defined way, and then optimizes using Gauss-Newton optimization, which is it uses the analytical solution, the analytical derivative of a least squares cost to get to the local minima in fewer steps. And I feel really that the method itself it seems quite complicated. It's working by learning displacements in the tangent plane and then projecting them back down to the sphere. But ultimately, it's those two things, I think, that are the main contributors to making Spherical Demons better than FreeSurfer. So it runs a whole lot faster because the optimization is quicker, and it's smoother because it's doing it in a coarse-defined way. And so if you're interested only in folding-based registration, Spherical Demons, until probably new MSM, was the best. MSM and Spherical Demons perform comparably in terms of the alignment, but Spherical Demons was much faster than the original implementation of MSM. Probably not true now. But again, Spherical Demons, the whole reason it's fast is because it uses Gauss-Newton optimization, which assumes that you've got a mean square error cost, sum of square differences, and sum of square differences makes a lot of assumptions. It seems the only difference between your source and target data is Gaussian noise. And this is rarely used in volumetric registration for a number of reasons. And as a result, Spherical Demons didn't actually perform very well on myelin-based registration either. So this is looking at the tiny little pockets, I can't remember what they're called, in the myelin. And we spent quite a lot of time obsessing over these. And you can see that MSM is doing a much better job. So fundamentally, continuous optimization was not good for myelin maps and fMRI. And my reason for that, I think, my intuition based on many runs, is that it's because they're much noisier data sets and you're much more likely to get stuck in local minima, and they're not suited to continuous optimization. So at some point down the developmental pipeline, we thought about trying discrete optimization, which was already used by Ben Glocker from Imperial for volumetric registration. And this just changes image registration. Basically, a discrete optimization means rather than allowing a control point to move to any location, it can go anywhere, it's just stepping in the direction of the gradient. You're giving it a fixed set of target locations that it can go to. It can't go to anywhere else but these. It's actually a label assignment problem. It's like a multi-label classifier or a segmentation problem. It's exactly framed in that same way. So you have to figure out which label is represents the best step for that control point. And in order to do that, you again balance the data similarity term and the regularization cost. And then what you've got is a combinatorial optimization problem. You've got to figure out what combination of labels across all control points works best for improving similarity balanced by your regularization penalty. That's it. So the data term is a unary potential. You're moving patches of data centered around the control point and looking at how well overlapping they are for each proposed move. And your regularization cost for your original MSM was a pairwise potential, so a first order, let's compare the displacements of neighboring points, but then that is relatively weak regularization. So in MSM, the second MSM paper, we moved to a higher order regularizer. So yes, just to re-summarize what I already said, discrete optimization, essentially the reasons why it's good is because you have no constraint, you have no constraint on what similarity term you use, similar to Gauss. Spherical demons has to use sum of square differences. Discrete optimization, it can use anything. You can use anything for either of these two terms, so that's flexible. But I think the key benefit is that it's so robust to local minima because you can just jump over them. And it was optimized. It was relatively fast, but not really that fast, which is where Renato came in, and it was optimized using a discrete optimization algorithm called FastPD, which means fast prime or dual. And I'm not going to go into details about that, but you can look into it in this paper by Komodakis in 2007 if you're interested. Okay, so the original pairwise method. So this is a Markov random field. And Markov random fields, most of the solvers only exist for first order Markov random fields, which means that you can only have pairs of labels, which means that you're learning first order regularization. In other words, you can only penalize movements of pairs of points, not triplets of points. So you can't do aerial corrections, you can only do distance-based corrections. And this has never been good for any volumetric registration algorithm ever. But it was worth the cost because the control point grid cost-defined registration afforded some smoothness, and the discrete optimization allowed us to deal with the noise in the data. So the pairwise regularization, all it's basically doing is it's comparing distances, but the distances are actually rotations of points, control points across the surface. So every time you move, you prescribe a proposed move, actually what it's doing is rotating from its current location to another point on the sphere, and you can aggregate those over displacements by combining the proposed move with historical moves, and that's it. So it's just a distance-based comparison of how far have they rotated away from each other. But this wasn't great. We had certain problems, especially when we went to multimodal registration. So in a second paper, we used this method called higher order clique reduction. This is an algorithm that takes these triplet cliques and reduces them to pairwise cliques so you can optimize them with FastPD. That's it. And that allowed us to use a more complicated regularizer term, which was inspired by biomechanical models of cortical growth. So it's a hyperelastic strain penalty. It looks like this. The reason we chose this was because it was motivated by a desire to build longitudinal models of fetal or neonatal cortical growth. But as we'll see, it works very well for a wide range of different tasks. Okay, I'm going to stop talking too soon, promise. So I noticed that no one else was talking about MSMUlc. So I'm going to talk about MSMUlc. So MSM was originally designed to do multimodal aerial alignment. And that means that we're driving registration using a combination of features that align with what we believe to be the underlying cytoarchitectonic or aerial organization of the brain. So we had 40 core scale resting state ICA maps, and we had a myelin map, and I think the aerial parcellation also uses what they call visuotopic maps, which are sort of hand engineered veteranatopic maps. So you have a number of different maps. They regenerate a feature vector at each location, and we're trying to improve the overlap of those feature vectors in the source and target mesh. And when you do that, as we've already seen, you get significant improvements in the overlap of task activation. So all good. And I don't know the extent to which everyone else is going into the configs, but seeing us been talking about the method, I just wanted to touch on some of it. So when you look at an MSM config, each one of these columns represents a different step in the registration. So you start with what we called regrettably affine, but what we actually mean is a rigid rotation to get things broadly into alignment with the target mesh. And then each one of these represents a different step in the course define registration. So you start with a low resolution icospheric control point grid of ICO2, which is about 162 vertices, and then you incrementally step up. This is the resolution of the label grid, so where the labels are placed relative to your control point grid. So usually that's 2 resolutions higher than your control point grid. Nobody ever really changes that. Your data grid is the resolution of your image, so that really generally needs to be high resolution. So you're actually comparing the data at relatively, something relatively close to its original resolution, so you can see all the features. Some of the influence of your choice on that will be like the higher resolution you go, the longer it will take to do all the calculations. This tells you to run MSM pair, which you really almost never want to do anymore, unless you wanted to do some really quick prototyping. But MSM strain is now very fast with Renato's new implementation. These are smoothing panels, so you can take your data grid and you can smooth it so that you are accounting for the fact that you're really only you smooth away all of the fine detail and you only are interested in aligning the core scale features to start off with, and then you reduce your smoothing. And this is basically really the only parameter that you need to optimize, which is the trade-off between the data similarity term and the regularization. Everything else can more or less stay as it is. These are the number of iterations you do per control point grid resolution level. They're low here for MSM pair, but when you go to MSM strain, we created like a new parameter, rescale L. What that does is it sort of jiggles the label space around, it offsets it so that actually you're sampling within each label separation. So it gives you a denser sampling of the space, but then you have to increase your number of iterations. And more importantly, MSM strain has all these extra parameters that go into the calculation strain. You can just leave them as they are most of the time. There's a fixed set for spherical registration and a fixed set for anatomical registration, and don't change them. That's it. Oh, for the HCP, the other thing you can consider is that you can, as well as running triplet-based regularization terms, you can run triplet-based data similarity terms, which are looking at the overlap of triangular faces rather than patches around each control point. And they purposefully configure it differently. This is how Matt's thought it works best. So he, instead of applying smoothing but using the data at full resolution, he applied no smoothing and downsampled the data. In the end, they have more or less the same effect. And this is the variance normalization term, which means that every single feature is normalized to mean of 0, standard deviation of 1, which means no one map will override the others in any calculation. And that's it. I will stop talking. Oh, I forgot to put a photo of everyone in.
Hello, everyone. Today, I will talk about how to perform MSMSulc registration in neonates, children, and adults, as well as template generation, especially neonatal template generation. Here, MSMSulc, it means the registration is optimized for so-called depth features, and as we all know, to compare, group comparisons. Typically, we need to align their brain to a common reference template to make sure we are comparing the same anatomical regions across different individuals. Here, running the MSMSulc command is pretty simple. All we need is individual native space mesh and data as input, and as well as a reference template mesh and data. So here the key point is to select the right reference template. So when we select a right template, there are two considerations. The first one is the template should reflect study population demographics. Here, for children and adults, typically we use one single population average template derived from young HCP adult data because brain shape doesn't change a lot after childhood. But for neonates, we need a series of week-specific templates because for neonatal brain, here we can see the neonatal brain change very fastly in size and shape across different scanning week. And another consideration is we need to think about spherical projection. Typically in surface-based pipeline, this is a very important step, spherical projection. It's to project a cortical surface into a sphere for alignment, Emma has mentioned before. And typically this step can introduce distortions, shifting how the vertices are placed. The original DHCP pipeline use a method to avoid the extra software requirements, but it didn't perform as well as the free surfer method, free surfer adult pipeline method. And the new deep learning neonatal pipeline has fixed this issue by using a learning-based spherical projection method. It can reduce distortions. It means objectively, this method is better. However, we need to regenerate our neonatal surface template to match this improved method. And here we can see it's very clearly the spheres from deep learning pipeline and the original pipeline, they are totally different. Yeah, for neonatal template generation, there are several steps, and yeah, basically for our template generation, they are the same logic. And firstly, we need to align native space of all individual data to a common template space. Here I use HCP template space using a fun, or we can say region alignment. This will give us, the initial week specific templates, but these templates only capture the basic folding patterns of our brain, and we want to include more anatomical details. To improve this, we need to refine the templates using non-region MS spherical alignment, which will include more anatomical features. But during this iteration, something called group average drift can emerge. This happens when the spheres, when the sphere's vertices keep shifting in one direction over many alignment iterations. So to fix this issue, we use the inverse of spheres to remove the drift, the problem during the iteration. And finally, to achieve the left-right vertex correspondence, here I just want to keep it similar to HCP templates. And MSSulc registration was used to align the left and right hemispheres. So this is a basic steps for how to generate surface templates. And okay, now we move to MSSulc registration for neonates and children adults. For children, firstly, we can look at for children and adults. In children and adults data is very simple because there's only one single population average template. All we need to do is run MSSulc registration and then apply the registration deformation to native data. But for neonates, it's a little bit complicated because, as I mentioned before, we have a series of week-specific templates. Our final aim is to align each native space to the DHCP common template space, week 40. So here, we need firstly align the native space to the week-specific, the local DHCP template space. And then we can combine this registration deformation with the week-specific to week 40 deformation. This will give us the transformation from native space to week 40 common template space, which we can use to resample native data to template space. And typically, if you only focus on neonatal data analysis, your final aim is resample to DHCP template space. But if your case is to analyze neonates, children, and adults data into one combined analysis, you also need to resample DHCP space to HCP adult template space. Similarly, here we need firstly align the deformation from native to DHCP space with the deformation from DHCP space to HCP space. This will give us the registration deformation from native space to HCP space. we can still use this deformation to resample native data to HCP template space. Yeah, and here please note that all this deformation concatenation are achieved by using WB command surface sphere project and unproject. And for when we are applying deformation, we can use WB command surface metric and surface resample and metric resample. If you want to look at tutorials about how to use this command, you can look at Saga tutorial on YouTube. Okay, next I will present how to run MSSulc for children and adult data and for neonatal data, and how to QC the result of registration. No, yeah, just look at the graph, but a very brief introduction about the. Yeah, here we can see this is a registration code for children and adults data. Here you mainly need to define your template and configuration path. And if your input folder is HCP style output folder, you can easily use this, run this code for registration. And this is a script for neonatal registration. Here, the first step is the rotational transformation. We need to transform the sphere space to FSLR space. This is the first step. And then we can run MSSulc to align native space to weak-specific space. This is the main difference between neonates and children, adults. Following this step, we need to concatenate MS deformation from native to weak-specific space with deformation from weak-specific space to DHCP common weak forte space. And finally, we can resample the native data to template space. Yeah. So this is a script. This is MSSulc registration, and about how to QC the registration results, we can see here. Just use WB view. Yeah, this is we can see this is a HCP template SulC, and this is individual aligned individual data, and then we just click. We can combine. We can quickly check the quality of registration. Everyone is nice. Yeah. Here, typically, I will just firstly click the major, just like the primary, our primary brain cortex gyrus, the precentral and postcentral, and then yeah, this is a template. We can firstly select the major, vertex of the templates, and then project the individual data to see this vertex, if this vertex represents the same anatomical structures. Yeah. Yeah, they are the same, the precentral and postcentral gyrus. For the configuration file, here I just put some useful links, the configuration file and how to install new MSM and HCP templates, and the tutorial about how to use WB command surface resample and metric resample here. Emma have introduced a configure, how to set a configure files in many details. So yeah, basically that's all.
Well, now I'm going to talk about another application of the MSM. which is it can enable the registration in a hierarchical framework. And the idea of the hierarchical registration is very crucial for the accurately align of any kinds of cortical features, not only for the cortical folding, which I'm going to present here, or maybe in the future present to the functional features. So let me first motivate this by why doing this instead of the conventional image registration approach, which seeks to align those highly variable brain images to a population average template that is smooth and lack of detail. So cerebral cortex is highly convoluted, is very complicated in terms of the folding patterns with huge individual variabilities. Well, some of these variabilities such as the difference in sulcal length, the orientations, the locations can be solved by conventional registration approach. There are some other variabilities such as the branching, splitting, connection to the surrounding folds or existence of an extra fold. This cannot be solved by the conventional image processing method yet, because these conventional methods can assume the diffeomorphism, which might mean that one folding pattern can be smoothly transferred to another folding pattern. But you should assume that it's without breaking any topology of the cortical mesh. And here's one example. If this one existent of the extra fold here and here, the extra fold here, which is not present in the population average space, is forced to match this template, that is lack of this detail, and cause the registration to squeeze or stretch the patterns heavily just to match the template. See, here is trying to squeeze this into nothing, just trying to match the smooth template, and this is not biologically plausible anymore. So instead of directly register everything to a population average level, the idea of a hierarchical registration framework is to register the most similar patterns together, and the most similar patterns together, then gradually register the less similar patterns until they reach to a top and common space. By doing that, we can align the most often ignored detailed patterns in the conventional approach, first in the bottom of the hierarchy. So the overall pipeline works by pairwisely measurement of the similarity of the cortical folding and clustering of the cortical folding patterns, template generation, then bring the hierarchical alignment to the population average space. And this tutorial is going to explain how this works step by step. So we first need a large data set, which is sufficient to capture most of the cortical folding variabilities. In this case, we are using all the 2,220 hemispheres in the HCP data. The framework starts by measurement of the pairwise similarity using a fast deep learning based surface registration method called DDR here in the bottom. And these can fastly align all the hemisphere to all the other hemisphere in this data set. And it is also separately calculating the similarities in different regions of the brain. We divided the brain into 3 different lobar regions, use the DK atlas. Then with the pairwise similarity, we can use the hierarchical clustering to create a dendrogram that interconnects all the hemispheres in this dataset from the most similar to the most different. Then we threshold the dendrogram at the level of 30 clusters for each lobe to parse the variability into a set of discrete example clusters. The number of the 30 is chosen to balance between capturing sufficient cortical variabilities that closely match to the Ono atlas, which is the most detailed textbook to record the cortical folding variabilities from the postmortem brain. And then these example clusters are unveiled by template generation. This is done using the MSM registration. The detail of it might have been introduced by Kaili, but here is some changes of it, we first take the average of the example in one clusters, and we average them as an initial template. Then MSM is repeated over several iterations to update and refine the sharpness of these templates. And the first iteration was done using the circle depth to roughly get everything into the same space, and the rest of the iteration was using the curvature to take advantage of these finer details. So the final templates here are capturing cortical folding patterns, different from each other, and it's very sharp. This step is sufficient for you to align the individual hemisphere to one of these cluster template. But what we want to do is to find a way to register an individual finally to a common space, which now is not working here because each of these groups are having their own template. So the only way to do is that we need to find a way to register the individual clusters finally to a common space. This will involve aligning the cortical templates between the clusters. And to do that, we first generate the intermediate template by aligning and average pairs of the cluster templates following the dendrogram until they reach to the top population average space. The pairs of the templates are registered in both directions. The forward and the backward registration are averaged. So we average the forward registration and the inverted backward registration to avoid bias. Then we incrementally align each of these templates through the template hierarchy. This part is done using the workbench command through concatenating the registration steps. The registration steps that these warps from the first templates to the higher level templates to the top of the template, concatenate them together, then do a one-shot resample from the bottom to the top. And then now these templates are in the top space. We can directly register the subjects belong to these clusters to the top space, finally bring them to a common space. And this might seem a bit complicated, but doing the registration in this way can help us to align the details of the folding pattern in an accurate way. Here is a comparison between the conventional method and the hierarchical registration, taking the frontal lobe clusters as example. So the registration quality is showing by the circle depth and the curvature, these two features, the mean and standard deviation of them. In the conventional registration approach, we register the subjects to a population average template that lacks the folding details. So the cluster templates that's supposed to be very different from each other actually here look very similar, regardless of the circle depth or the curvature. They all look similar to each other. And also the registration arrow is actually great in places that apart from the major folds. The major folds are primary folds that have less inter-individual variabilities. And if we have a look at the hierarchical registration result here, we'll find that so the clusters, they are looking like quite different from each other now. They reveal more details, and also they are sharper in the presence between compared to the conventional approach. We can also see that in the standard deviation map, it reveals less registration error in these high variability areas compared to the conventional method. So note that both of the methods, they are having the same level of area distortion. So we're not pushing one better than the other one. It's a very fair comparison. Okay, so now don't forget that we are doing this separately for 3 different lobar regions, and we still want to combine this registration together in one hemisphere. So to do this, we make use of the cost of function weighting option in the MSM. It allows us to input a weighting mask to the moving and the target meshes, and this will allow us to control which part of the brain feature contribute more to this registration, because we give it a mask. And the mask is not like a binary mask. You can put whatever like values you want. For me, I put the DK Atlas derived the frontal lobe, parietal lobe, and parietal occipital lobe and temporal lobe, but I smoothed the margin a bit because I don't want there is a sharp change between like the place I want to focus on, the place I don't want to focus on. It will affect the registration. So our strategy is to register one hemisphere lobe by lobe, provided these three different masks. And one difficulty here is that when we do registration here in one lobe, because of the regularization term to keep the registration smooth, it will affect the other regions. Mostly the margin regions that like our neighboring regions are like affected by this iteration. So once you fix this lobe, then you do the other lobe, it will change the area in the previous lobe. So the way to overcome this difficulty is to still take turns to register these three lobes to three different separate templates, but we do it, repeat this for three different resolution levels from the coarse to fine. The coarse is ICO 2 and the fine is ICO 4. And in this manner, the region that are affected before in the previous level can be fixed in the next level in a finer scale. So we correct it back in the next level and we correct it back in the next level. And in the last level is ICO 4. It's actually very small patches. It won't affect much of the surrounding area. So this is fixed here. And in this way, we keep the cluster level details shown in these slides, but still we bring them to the common space so that we can do some downstream analysis and compare the individual subjects from one to another across these different clusters. To apply this to the other data set is a much easier approach because all we need is to register the new data set to the 30 template at the top common space that we have already defined using the hierarchy. And then we can choose the most similar template. After that, we can go through these lobe by lobe registration to put the entire hemisphere into this common space. And and this is some other applications in neuroscience that this hierarchical registration can benefit. Since this hierarchical registration enable us to look into the cortical variability in a finer scale, we can also make it possible for further investigation, such as the structural asymmetry and the folding function relationships. But since this tutorial is focused on the method, I will just we can discuss this in the future. That's it.
Okay, so just a quick background because most of the previous presentation already covered this. So as Emma and Yourong said, that spatial normalization to global average template can cause misalignments, especially in outlier examples, and because of the major differences in folding and functional activations, and it reduces the sensitivity of studies. So one solution can be to cluster subjects that share common shape variants and shape or functional variants that Yourong's been doing essentially, and align these subjects within a group with each other in a kind of a groupwise registration method without any template, so no template bias wheel involved, and this will cause a better alignment, hopefully, and also improves the sharpness of population average templates. So this is what we expect. And we recently implemented this, about the hierarchical clustering. This is one way to cluster subject. Obviously, there are different ways, and how groupwise MSM works is really not important how you cluster your subject, so it's not important, but one way to do it is cluster hierarchically, just introduced by Yourong. Okay, and we recently implemented Groupwise registration or re-implemented Groupwise registration into the MSM software. It was in it, but it wasn't really working in an efficient way. So we reworked the entire MSM software to work more computationally efficient. And now it is possible to do Groupwise registration. And this is just a high-level outline of the entire method, where we basically, I start this presentation. So where we actually align subjects with each other within the group without any template. So basically what we do is to move the control point grids and along with it all the sampling points on a spherical mesh. But now we are not comparing the individual data to a template data, but we are comparing pairwise with each other across all the subjects within the cluster and calculate the data similarity term according to this. After that, the regularization term or the triplet cost is calculated on all triangular strain for all labels along all meshes, and this is done individually, so all of the individual meshes assigned to the subjects. And then we do the optimization just we did in the regular MSM using a higher order click reduction and then fast PD. Then update the control point grid according to the optimal setup of the label choices and do this until convergence. So ideally when we find the global minima of the optimization process. Okay, and then we have the output deformation for all the subjects. If you want to go to the common space with all the subjects to do a population level comparison, you can do a cross group registration, but in our case this is optional, in my case at least. So you can create generate group means up along the hierarchy defined by Yourong's method by registering all the subjects to the other template of the parent node and iterate this process until you reach the top level here. And by then all your subject will be in the same space, so you can do population level comparisons as well. In my case, this is done with a typical new MSM registration process. So basically you are registering this subject to the other group template within the same level of hierarchy. Okay, so after this short and brief introduction, I will show you a few steps how to do this groupwise registration on clustered HCP young adult data. At the moment, we only have folding, Sulc depth and curvature. We do this in a kind of a multimodal way, so both Sulc depth and curvature plays a role in the registration process. I think we will do on function later. So theoretically, the groupwise mode of MSM can do function as well in a multi-channel way, but I just only tested briefly, haven't done a deep analysis on that. Okay, so what's the process? We align subjects within a group with each other and then do some post-processing and generate group averages after that. Okay, so what you need for that, first you need a list of subjects and groups. This is the output of Yourong's hierarchical clustering. So here the first is the line number, so just ignore it. The second is the subject and hemisphere ID and the number of node or group where the subject or hemisphere resides in. You can also set up a configuration file. This usually follows the general MSM registration config file. The major difference is that you probably need to increase the lambda between 0.2 or 0.5 depending on your work to be done. So it's a bit higher than in typical MSM, but the rest is basically the same. You also have a multi-thread option if you are interested in it, or you can use a multi-thread setup. Okay, we provide a script for this, and you can set your input folders and working directory and clustering files. Just really quickly, in this first part, up until this, we are just generating a few files that the Groupwise MSM needs to work with, a list of input files and a list of input meshes. So it's really administrative things. And then we invoke or call the new MSM process to do the registration itself. And then we do a de-drifting phase. As Kaili mentioned, we are experiencing some drifting within a group. So this script does all the de-drifting for you, and then at the end we are generating all the Sulc depth and curvature, and so the merge and the mean files and the standard registering a very small group of in this case free subjects. It's not really meaningful, so registering in a free subject in a Groupwise method is not very meaningful, but Groupwise MSM now can do 250 subjects, so you can register 250 subjects in one Groupwise process. At least that's the highest number we tested on. Theoretically, it could do more, but we are experiencing some problems with FastPD. FastPD is not that fast when you have a lot of terms in your equation, but it's really a high stress for that as well. But 250 subjects should work just fine. Okay, so I start this registration. You will see a lot of debug messages, so you can save these into your log files. And for free subjects, it should finish like in a few minutes. But until then, I will show you the results because you can visualize that as well using like the workbench view. I'm just using a random anatomical surface. I don't know whose anatomical surface is this, but it's just for the sake of example. And then you can visualize all the results that the Groupwise MSM outputs. Yeah, sorry. It will be fine. Just, yeah, I need to set which group I want to visualize. Okay. Yeah, so these are all the files that is the output of the Groupwise MSM method and the script that is provided for you. So this is like the mean curvature. And you can also visualize the standard or the mean Sulc depth, and you can check all the subjects here. Okay. So these are the free subjects that are in this group. Yeah, So also have the standard deviation maps, and yeah, pretty much this is what you have. I also want to show you the files. This is the clustering file where you have the list of subjects with the clusters. This is your input. In our case, it's as I said, the ACP Youngdale dataset, and the program will generate you the output here, the surfaces and the data. And here in the results, you can see the what I just visualized in the WBView program. Okay, and yeah, it's supposed to finish by now, but yeah, it can take a few minutes. So yeah, this is it, and thank you.
So most people before were talking about registration in the case of registration to a template space, most cases that we heard on Wednesday, although Emma talked a bit about registration overall. And so this is where we register individual metrics to a common space, and it's particularly suitable for group analysis. And another use of surface registration is longitudinal registration, where we can register 2 time points or more, obviously, from the same subject. For example, in this case, just have like one subject was scanned at 36 weeks and then another at 42, and then register one subject to the other to get a vertex correspondence between the 2 scans of the same subject. This is particularly suitable to study longitudinal patterns. I mean, either growth in this case, or I mean, obviously can also use this for along the entire lifespan, so you could also use it for neurodegeneration, etc. And it's important to note that, I mean, obviously Emma mentioned when she was presenting her work about how we want to ensure that registrations the wraps are smooth and but this is particularly important in longitudinal registration in the sense that there's an actual underlying biophysical process behind like what is actually happening in the registration. So this is actually a physical process where the brain is actually, well, in this case, growing and folding, rather than us just using different subjects where this doesn't really happen. And so the goal of AMSM is to incorporate the anatomical surfaces, and by anatomical surfaces, most often mean we use either the white matter or the mid-thickness surfaces to regularize the registration. And like I mentioned, this is particularly suitable for intra-subject longitudinal registration, where we have multiple scans of the same subject. And so here so the framework of MSM is the same as like what Emma described. And it's important to note that here the warps are still inferred between the spheres and not in the actual surfaces. So the registration itself, how we deform the points on the spheres driven by the metrics are done the same way as what Emma described. But then we can infer an anatomical warp by projecting the topology of the moving sphere to the target anatomical surface to create a deformed anatomical surface, which then is going to have point correspondence to the target. This is where we actually calculate the strain regularization that Emma talked about, and I'll just talk a bit more next. So the registration itself is done on the sphere, but then we use the anatomical surface to regularize the registration. And so just like Emma talked about, in the case of anatomical MSM, these deformation gradients are calculated between triangles in the anatomical surface. For example, here we have the input anatomical surface and the deformed surface, and these are the vertices that correspond to each other. And we can calculate a deformation gradient, or just by the affine transformation between the triangles, between the input and the deformed anatomical surface, which then is used to calculate the hyperelastic strain energy regularization, which is then applied as the in the regularization term of MSM. And it's important to note that this strain hyperelastic regularization is actually based on models of brain deformation and growth, where R corresponds to changes in J corresponds to changes in area, and R corresponds to changes in shape. Yeah, shape. Thank you. Yeah, so that's basically the main difference as how AMSM is implemented. In terms of running AMSM, so most often the data that we use for running AMSM is going to be the either the curvature or the suckle depth metrics, although I assume we can use still any, including multimodal. And in terms of running it, the same applies as to normal MSM, other MSM, in terms of the input and reference meshes data. And then the difference here is that we need to also include the input anatomy and the reference anatomy. And in this case, for example, if we just have like one just random subject that has two sessions, then obviously it's going to be session one for the input, for example, and session 2 for the reference. So in this case, we are registering session 1 to session 2. And in terms of the configs, so Emma also already talked a bit about like the config file for MSM in general, including MSM strain. In terms of anatomical MSM, the main difference here is going to be the grid size for the anatomical surface, which is often just left the same as data grid, but yes, that's something you can change. And then obviously also the this reg option here is to specify that we want MSM 5, like Emma also had talked about. And then this bulk modulus shear models are going to be the what is going to be here in the strain energy. And so the difference here again is just the anatomical grid. We also need to define how many vertices we want for the anatomical grid. In this case, just show an example where it's the same as the sampling grid, and these are just examples of how it looks like at each step of the registration. And so in the original, so AMSM was first proposed in the multimodal surface matching with higher order smoothest constraints paper. And in this paper, also did some tests, some tests to show that how AMSM strange provides improvement in longitudinal registration by registering 10 preterm subjects with 2 scans at different time points. And then you can see that compared to other registrations where the only like spherical techniques and deformations are used, there's in AMSM in comparison has much smoother so areal differences across the brain than methods that are, obviously MSM is still a spherical method, but methods that are strictly spherical. And so by regularizing on the anatomical mesh instead of just the spherical mesh, we can have a significant improvement in longitudinal registration, and which shows can provide better analysis of the real trends of how the brain is growing. So in this case, they calculated the areas where the expansions are significantly above 0 between these 2 time points, and you can see that AMSM provides much interpretable and reasonable maps. And then AMSM has also been used by Cara Garcia, who was also in the first AMSM paper, to study cortical growth in 30 preterm infants that were scanned 2 to 4 times during the period for 28 to 38 weeks post-menstruate age. In this case, so for some subjects, they had multiple scans, so they did multiple registrations from time point 1 to 2, 2 to 3, and 3 to 4. And then, so individual registration was done using AMSM, but then just to note that group analysis was still done by registering to an atlas, which was done with just MSMSulc. And so AMSM was used to calculate continuous maps of cortical expansion, and they were shown to provide more fine detail of where cortical folding is happening compared to only ROI studies. And in this paper, so this is on the supplementary material of this paper, but Cara also performed a bit more in-depth study of the effect of the actual biomechanical regularization, so the strain regularization. First of all, she showed that that having the regularization doesn't actually negatively impact like the actual alignment of folding. So just showing like she did like a small test by deforming like a smooth sphere to the sphere with some folds, and you can see that the actual folding does stay in the same place, but the relative expansion maps are much smoother when using regularization than when not using, but they still are able to deform in a way that actually matches the folding patterns. Another thing that's important to note, and then I also use in my methods, is that it was shown that in order to reduce bias, it made sense to do both forward and reversed MSM to then get unbiased warp between the scans by registering, so not only registering the younger to the oldest, but then also vice versa, and then averaging the maps, because the direction of the registration is going to have some impact, especially when there's less folds in one surface than the other. And then I applied the same kind of method, in this case by registering, using MSM to register both subjects that were scanned in utero, so fetal, and then after birth, and then subjects that were born preterm, that was born shortly after birth, and then anatomical given age. And I applied the same kind of methodology as in Cara Garcia's paper, where we register first scan to second scan, and vice versa, and then average the results to get the cortical expansion maps. It's important to note, because I had like the fetal scans were quite young, some of them. I did have to do some optimization on the regularization term lambda weight factor, which, just to show that this is kind of a parameter that needs to be optimized. And in a case where I had very young fetal scans and then to neonatal scans, there was a big difference between the folding patterns, while if they are closer to each other, it's like less difference. But I had to find, I found a lambda that was suitable for the entire cohort. Could obviously also do it separately depending on the case. Some other cases, so again, longitudinal registration. This is also a paper by one of Cara Garcia's students, where they looked at cortical expansion here between neonatal and surfaces at 9 to 10 years old in children born very preterm, and comparing to children born in full term. And so again, they used AMSM to register the neonatal to the surface at age 9-10 to calculate patterns of expansion, which then were the subject-specific expansion maps, again, were calculated by MSM, and then they were registered to a template using MSMSulc. And they showed that children born very preterm experience increasing cortical expansion in these highlighted regions. And then finally, I found this was funnier to share than anything else, but just to show that IMSM has also been used on non-human brains, and in this case, also by Cara Garcia. She looked at fit ferret brain development. So in this case, they actually had 6 time points per subject in 10 ferrets. So from day 8 post after birth to day 38, because actually most of the folding in ferrets occurs after birth. So they actually did registration from all time points to the last time point. So from this to this, then this to this, this to this, and then to calculate the in between, they just subtracted, which is also important to note that because in the ferret there's also only there's less folding than in the human brain, because there's only primary or maybe secondary. I don't know if there's secondary folding in the ferrets actually, but because there's less, there's also obviously the registration is gonna, even between the very young and the oldest, is gonna be easier to predict than for the humans. And this is just to show another use case, and that's it.
Watching this bit, I do think it's important to point out that the deformed anatomical surface has the exact same shape as the target anatomical surface. This is enforced because it's a spherical mapping. So unlike in volumetric mapping, you might not have complete overlap. It's the same surface, but with the mesh topology of the source. Because I think that's confused people before.
Yeah, including me when I first started using it.
So there you go.
Yeah. Okay. Thank you. I don't know if anyone has any questions.